Hi, my name is Qiyuan Han. I'm a graduate student at the Tretyakova lab. And today I want to show you how to do the MIDIP-seq. MIDIP-seq is an abbreviation of methylated DNA immunoprecipitation followed by next-generation sequencing. This technique is used to map where the methylated DNA is located along the genome in different conditions. So the first step of this procedure is to extract the DNA from the tissues or cells that you are interested in. And then the step 2 is to fragment the DNA. This is done by usually by the University of Minnesota Genomics Center. So briefly, you just send the extracted DNA to the UMGC and then let them know the conditions that you want to fragment your DNA. And then after that, they will do a size check for you to show you that the DNA is fragmented to the ideal size that you want. And then they will send the fragmented DNA back to you. And then the step 3 is to do the immunoprecipitation of the DNA that has the modification that you are looking at. Next, I will show you how to do this immunoprecipitation of the DNA, interesting to you. So every time when you work with the next generation sequencing protocol, it would be ideal to clean out your bench first with the DNA off reagent. This will degrade all the DNA that may potentially present on your bench before, and it will prevent any contamination DNA that coming from other resources other than the DNA that you are interested in. So you just spill the reagent on the bench and then wipe it with the Kim wipe. And then I would wait for like 5 minutes for it to clean up the previously presented DNA. And as I said, from step 2, you are supposed to get the fragmented DNA from the UMGC. And I assume you have 2 conditions, that is the control DNA, and there is also a DNA from the treatment group. And for doing this, it is always recommended to have a no template control. This is like a mock sample that you are doing exactly the same as your actual sample, but without putting the actual DNA in it. So this sample will help you control if there is any contamination DNA coming from the process that you are working on. These 2 tubes has the fragmented DNA from the UMGC, and we need to make a mixture of the immunoprecipitation. So this is done by mixing 3 things. The first is the fragmented DNA from the UMGC, and the second is the immunoprecipitation buffer, and the third one is the specific antibody to the modification that you are interested in. In this case, we are interested in the antibody that is specific to the N6-methyldeoxyadenosine. So you just mix them together. First of all, I would add the nucleus-free water. So for the nucleus-free water, I usually use the ultra-pure water that comes from the RBS reagent kit, or you can also buy it separately. Anything that is nucleus-free and ultra-pure can be used in this protocol. So you just pipette the amount of nucleus-free water that you need into the tube. And next, the IP buffer. And then the truncated DNA. And at the end, the specific antibody. And you do the same thing for the treatment sample and the no template control. When you add all the things that you need for this reaction, you just do a finger click, and then you brief centrifuge them down. So you just leave the reaction mixture on the rotator at the cold room, and then let it rotate overnight at 4C. So the next day, we are going to do the IP with the magnetic beads. So for this step, we need to first of all get the beads ready. So for 5 micrograms DNA for each sample, you need 15 microliters beads. You just take the beads and then put it in an Eppendorf tube and then put the Eppendorf tube in a magnetic stand and wait for about 1 minute. So the beads will be magneted to the back. And after 1 minute, you will take the supernatant out, and then you get it back to the rack. And then after you get rid of the storage buffer of these beads, you want to wash it with 1 ml of the 1X IP buffer. Add the buffer to the beads, and you can do a brief vortex. And then you put your beads onto the magnetic stands again. Wait for about 1 minute, and then you took the supernatant out. Now the beads is ready to use. And now you can just take the reaction mixture from the cold room and then add the reaction mixture directly to the beads. And now this is the tube with the IP mixture you did yesterday night, and then with the beads that we just prepared. And now you put this tube on the rotator at the cold room again and then rotate it for 2 hours. So in step 4, we did the immunoprecipitation with the beads. So after the 2 hours rotation at the cold room, we took the samples back. And now we are doing the step 5, which is the washing step. Basically, this step is to wash any non-specific DNA that is accidentally bound to either beads or if it is just a non-specific binding. We will wash it with 1X IP buffer 6 times and then 3 times with the 1X IP buffer with extra high salt concentration. This is the tube that we did with immunoprecipitation with the beads. And then first of all, you just put it on the magnetic stands again, and then wait for about 1 minute or until the supernatant is clear. And then you took out the supernatant and add 1 ml of the 1X IP buffer. You did this with the rest of your samples. And you then put it on the rotator again. And then rotate for 5 minutes at room temperature. After this is done, you want to briefly centrifuge it to collect all the solvent. And then put it on the magnetic stand again. Wait for 1 minute or the supernatant to be clear. And then you took the supernatant out again. Do this for the rest of the samples. Put it back on the rack. And then basically repeat the washing step as what I just showed you for a total of 6 times. And then after the 6th time is done, you use 1X IP buffer with 300 millimolar sodium chloride. So this will enhance the washing power. And then you wash as what I just showed you for another 3 times. And this is the end of this step. So after all the rigorous washing step, we are going to do the step 6, the DNA that has been bound to the beads and the antibody. So the way we do it is we use a high concentration of the N6-methyl-DA nucleotide to compete with the DNA that has been bound to the antibody. Basically, you just add 100 microliter of this 2.6 millimolar N6-methyl-DA nucleotide to the beads after the last step of your washing. Pipette mix it, and then let it rotate on the rotator for 40 minutes at room temperature. And then after 40 minutes, you put the tube on the magnetic stand. And then you collect the supernatant. Collect the supernatant into a new tube. The supernatant is supposed to contain the DNA fragments that has been competed out by the high concentration of the N6-methyl-DA nucleotide. And then you repeat this computation step 1 more time. So after the second computation, you combine the supernatant to the same tube. So this tube gonna contain all the DNA fragments that has been fished out by the specific antibody. So then we go to the step 7, where we will precipitate the DNA fragments. So we do this by basically the ethanol precipitation. So you want to add the following things to the supernatant that we just collected. First of all, the 100% ethanol, the glycogen, which will help precipitate the DNA, and also the 3 molar sodium acetate, pH at 5.2. And then you leave this tube to the -80 overnight. So after the overnight incubation, we will continue the DNA precipitation the next morning. So the first step is to centrifuge the mixture at the maximum speed at 4C for 25 minutes. And then after this step, your DNA is supposed to form a pellet at the bottom. And then next, you want to wash the pellet with 1 ml of 75% ethanol, and then centrifuge at maximum speed for 10 minutes at 4C. And then you just remove all the supernatant, but leaving the pellet at the bottom, and then allow air dry for 2 to 3 minutes. And then after that, you will dissolve the pellet with 23 microliter of nucleus-free water again. You want to do a brief centrifuge and vortex. And now you want to quantify the concentration of the DNA that you immunoprecipitated from the original DNA by qubit. So after you got the concentration by qubit, we will be ready to move to the library preparation step. That is step 8. So then we went to the first step of the library preparation. The first step of the library preparation is the end repair. So basically, you will first of all transfer the resuspended DNA from this 1.5 ml Eppendorf tube to a PCR strap. So after you transfer all the resuspended DNA to this PCR strap, you just basically add the rest of the reagent that is needed for the end repair, as shown on the next slides. And then after you mix all of them, we want to do a pipette mix, because this step contains an enzyme, and then you don't want any fierce mixing action, but use a gentle pipette mix for 10 times to mix the mixture. And then you spin it down. So now the mixture from the end repair step is ready to be incubated on the thermocycler. You need to pay attention to the PCR strap that you are used. If it is a non-flat cap, you want to leave the rack with a non-flat cap on the top, and then you put your PCR strap in. So you want to close the lid. And then when you fasten it, the first time you feel like a resistance. This is the point that you need a quarter of screw. So this is a good stopping point to close the lid. And then you just open the program that you have set up. And now we are running the first step of the library construction, which is the end repair. I had a program named IPSeq1 that is for the end repair step. And then you pull this step up, you pull this program out, and then you just click run. Okay, I'll just click to run. Pay attention to the volume that you are doing. It's supposed to be 30 microliters. And the lead temperature is usually set to be 10 degrees higher than the highest, actual highest temperature in your protocol. And now it starts the run. When it reached the final step of infinite incubation at 4C, it means it is done. Now when you reach the infinite holding step at 4C, it is done. You want to click cancel in order to stop the program. And when the light is off, it's ready for you to take your PCR strap out. So while you are waiting for the thermocycler running for program 1, you can take the adapter out from the freezer and solder it on ice. Do not vortex it because it is very fragile. And at the meantime, you can also take the impure purification beads out and then leave it on the bench at room temperature. It is needed to be equilibrated to the room temperature. When the end prep program is done, we are ready to go for the next step. That is the second step of the library construction, adapter ligation. So for this step, you need to pay special attention to the ligation buffer because the ligation buffer is very viscous. You want to pipette super slowly. And then you just follow the instruction to basically mix all the required reagent into the tube and then incubate on the thermocycler again. And because this adapter is the circle one, after the first incubation of the adapter ligation, you want to add the user enzyme to cut the circled one to make it as a flat or blunt end. And then after that, we will move to the next step, that is the impure XPB's purification. So this step is basically just to clean up all the previous reagent, but recover the DNA that we want. You just follow the instruction to recover the beads. So the next step is to do PCR enrichment of the adapter ligated DNA. So basically, you just mix a bunch of reagents as shown on the instruction sheet. One thing that you need to notice is the PCR cycles that you want to perform. This is usually decided by the initial input amount. You can follow the instruction table as shown next. But if you are not sure, you can also do a qPCR quantitation to make sure you get the appropriate PCR cycle for the enrichment in this step. And then after the PCR enrichment, you basically do another round of impure beads purification to recover the DNA that is adapter ligated and enriched. And then after that, you will just quantify the library with the qubit instrument again to get a rough idea of the amount of DNA that you recovered from the whole library preparation protocol. And then so far, it is almost done. And then you can follow the instruction from the UMGC about how to submit your library, submit your sequencing library and get it sequenced.